Hello everyone, myself Pallavi Sharma. Here are some important questions about the chapter Cell, the fundamental unit of life. So we are going to discuss one by one. First question is common characteristic feature of plant sieve tube cells and most of the mammalian erythrocyte. So first option is absence of mitochondria, second option is presence of cell wall, third is presence of hemoglobin, fourth is absence of nucleus. So correct answer is absence of nucleus. Why not absence of mitochondria? This is the reason behind this. The erythrocyte loses its mitochondria at maturity because it has to maximize its space for hemoglobin and its main function is to transport oxygen. So that is why it loses its mitochondria at the later stages. While sieve tube cells, it is a phloem tissue. Its activity is controlled by the companion cell, which is another type of phloem cell, and its all energy requirements are completed by companion cell itself. That is why sieve cell do not possess any kind of mitochondria even in early stages. While the presence of cell wall is itself not a correct answer because RBC is a animal cell and sieve cell is a plant cell, so it possesses cell wall, while RBC is an animal cell, it doesn't possess any kind of cell wall. The third option is presence of hemoglobin. So only erythrocyte or RBC contains the hemoglobin because its main function is to transport oxygen. Fourth option is absence of nucleus. Yes, this is the correct answer because both of these structures or both of these cells lack nucleus because their activity of the sieve tube is controlled by companion cell, while RBC's main function is for hemoglobin, for containing hemoglobin and transport oxygen to the various part of the body. So D option is the correct for both. Next question is which one of these is not a eukaryote? So beta, euglena present in the group of protista, and protista are eukaryotic organism. Anabaena is a bacteria. They are present in kingdom Monera according to Whitaker's classification. Spirogyra is algae. It is present in kingdom Plantae, so it is a eukaryote. While Agaricus is a fungi, it present in a class Basidiomycetes. So they have asked which of the following is not a eukaryote. Euglena is a eukaryote. Spirogyra algae, Plantae, it's a eukaryote. Agaricus, fungi, it is a eukaryote. Apart from Anabaena, every organism present here is a eukaryote, so the correct answer is B. Next question is different cell have different sizes. Arrange the following cell in an ascending order of their size. Choose the correct option. So we have first option is mycoplasma. Mycoplasma is of 0.3 μm in size. Ostrich egg is about 15 cm in diameter. Human RBC is 7.0 μm in diameter, while bacteria is 1.1 to 2 μm in size. So if we are arranging them in a ascending order, the smallest cell will be mycoplasma, which is present in first. Then moving on to next, then other cell is bacteria, fourth. Then bigger than this is RBC, which is third option, 7.0, and last is the largest, which is ostrich egg, second. So correct answer will be A. Next question is, which of the following statement is true for secretory cells? First is Golgi apparatus is absent. Second is rough endoplasmic reticulum RER is observed in the cell. Third is only smooth endoplasmic reticulum. D is secretory granules are formed in nucleus. The correct answer is B. The reason behind is this, that Golgi apparatus involve in packaging and modification. B option RER, it involve in protein synthesis because it is associated with ribosome. So secretory vesicle, specifically the secretory cells, are responsible for formation of various type of protein and if a cell is responsible for the formation of various type of protein, then RER is involved majorly in protein synthesis. That is why secretory cells contain more of RER. Then third option SER, the smooth endoplasmic reticulum involved in lipid synthesis only, lipid or fat synthesis only. And D option, secretory granules synthesize in cytoplasm only. And they have written it is present in nucleus. So the correct answer is. Next question is, plasmid are extrachromosomal circular DNA molecule which have their own DNA, which have their own point of replication and can replicate independently. This is the correct answer. Second option is, which have their own point of replication but cannot replicate independently. Third option is, which do not have their own point of replication and cannot replicate independent of bacterial chromosomal DNA. And D is none of these. So the bacteria contain plasmid, and plasmid are extrachromosomal small circular DNA present in bacterial cytoplasm have their own ori. Beta, ori is origin of replication. So they have, this is point of replication from where they will start their replication. So they have their own point of replication and they can replicate independently. The second thing is, they have written they do not have, we have mentioned out they have their own ori. The third option is they do not have their own point and they cannot replicate independent. So they do not depend on the original nucleoid or the original bacterial DNA for their replication. So this option is also wrong and D is already wrong. So correct answer is A. So moving on to the next questions. So next question is, three of the following statement regarding the cell organelle are correct while one is wrong. Which one is wrong? So we are going to answer this. Lysosome are double membrane vesicle bounded of from Golgi bodies and contain digestive enzyme. This is the wrong statement because lysosomes are single membrane bound organelle. The endoplasmic reticulum consists of network of membranous tubule and help in transport and synthesis and secretion. This is correct. Leucoplasts are bounded by two membrane, lack pigment but contain their own DNA and protein synthesizing machinery. This statement is also correct because the plastid contain various pigment. Apart from that, one pigment is leucoplast, and leucoplast is responsible for storage of various nutrients such as starch, protein, fat, etc. And spherosome, these are single membrane bound organelle associated with synthesis and storage of lipids, specifically in plants. So all of these three of the statement are correct while A statement is wrong because lysosomes are single membrane bound organelle. Apart from this, all the details are given are correct, like they are vesicle budded from Golgi body. This is correct. They contain digestive enzyme. This is also correct, but they are single membrane bound. That is why correct option is A. Next question is, bacterial gram positive cell wall contains. So bacteria are classified into two types. There was a scientist, his name was Hans Christian Gram. He classified bacteria on the basis of their ability to stain, retaining power, into two parts. One are gram positive and other one was gram negative. So gram positive bacteria, they contain mucopolypeptide, while gram negative bacteria, they contain lipopolypeptide. Mucopolypeptide is mucilage plus peptide, amino acid plus carbohydrate. These are lipid plus polypeptide, lipid plus protein. Okay, so these bacteria contain 70 to 80% of mucopolypeptide, while these are rich in lipopolypeptide, so they contain only 10 to 15% of mucopolypeptide. So here in the question it is asked that bacterium gram positive cell wall contain, that B answer is correct, the 70 to 80% of mucopolypeptide. Next question is, mesosome of prokaryote performs functions similar to. One is peroxisome, second one is mitochondria, third is lysosome, and D is ribosome. So correct answer is D, because first is peroxisome, they are involved in fatty acid metabolism, that is breakdown. If we talk about lysosome, they are involved in digestion may be intra or extracellular. If we talk about ribosome, these are known as protein factory, so they are involved in protein synthesis, while mitochondria involved in aerobic respiration and result of aerobic respiration is ATP synthesis. So the correct answer is B. Next question, plasmid are extrachromosomal DNA present in, this is similar question. So these are extrachromosomal DNA we have learned. So these are present in bacteria only. Viruses are non-living particle until they are present in living body. These are non-living particle until they do not present in living body. Next is protista, these are eukaryotes. Then plant cell, these are also eukaryote, they do not possess any kind of plasmid. Only the viruses are the lining between non-living and living. The bacteria do contain these structures called plasmid, and these plasmid contain extra DNA. So correct answer is A. Moving on to next question. In animal cell, protein synthesis takes place in, one is on the ribosome present in nucleolus as well as in cytoplasm. So what a nucleolus is responsible for rRNA synthesis or ribosome synthesis. So this is not the correct answer. B, on the ribosome present in the cytosol as well as in mitochondria. So this can be the right answer. We'll move to the next option. Only in the on the ribosome, protein in the cytosol. This is not the correct answer. D, only the ribosome attached to the nuclear envelope and endoplasmic reticulum. Then what about the ribosome present in mitochondria and plastid? We have studied that mitochondria and plastid ribosomes are 70S ribosome, and they are responsible for some of the protein synthesis of these organelles. So this option is also wrong. So the very correct answer is B option, just because they have mentioned that ribosome present in cytosol as well as in mitochondria are responsible for protein synthesis. We have studied that cell have two type of ribosome. One type is organelle ribosome and second type is cytoplasmic ribosome. So organelle ribosomes are present in mitochondria and chloroplast, while cytoplasmic ribosomes, they may be free in cytoplasm or bounded with something. So mitochondrial ribosomes are 70S, while chloroplast are also 70S. These both are 80S ribosomes, and all of these are responsible for protein synthesis. All of these participate in protein synthesis. That is why answer is B. So next question is, chromosomes are stained with one of the following chemicals. So first option is light green, second is eosine, third is safranin, D is acetocarmine. We have studied that chromosomes are stained by, stain means color, stained by basic dyes, and these basic dyes can be acetocarmine, basic fuse chain, methylene blue, toluidine blue, etc. So here are the options given. That light green is used to study the histology of the cell, and this will never used alone. This will usually use with along with safranin or with eosin, used to study histology. Histology means the tissues or the cells, specifically the cells of the any organism. Second is eosin. Eosin dye is used to study cytoplasm because it is pale in color. Next is safranin. Safranin is used to study endospores or bacteria, and acetocarmine is basic dye. This bind with chromosome, so we can study the mitotic stages or mitotic chromosome by using acetocarmine. So the correct answer is D. Next question is respiratory enzyme present in following organelle. So chloroplast, no, because chloroplast is responsible for photosynthesis. Lysosome, these are responsible for digestion. Peroxisome, these are also responsible for breakdown of fatty acid. Mitochondria, yes, this can be the right answer. How? So we'll study about that. See, here is the outer membrane of mitochondria. The inner membrane is bit infolded. There are infolding of mitochondrial inner membrane. The inner mitochondrial membrane contain various structures, various protein, along with there are some structures So these structures are known as oxysome, and these are the proteins. These proteins are responsible for electron transport, and the result of electron transport is generation of ATP. Okay, and these proteins contains respiratory enzymes. So that is why answer is C, mitochondria. So the next question is, which of the following statement regarding the cilia is not correct? So cilia contain an outer ring of 9 doublet microtubules surrounding the 2 singlet. Beta, please see this diagram. These are 9 microtubule doublets. These are known as peripheral doublets, and these peripheral doublets are total 9. Okay. And next is, they have said that they are surrounding the 2 singlet microtubule. These are 2 singlet microtubule. These are also known as central singlet. So this statement is correct. Next, the organized beating of cilia is controlled by fluxes of calcium across the membrane. So this is not correct, because the organized beating of cilia and flagella is controlled by mitochondria itself, not by the metabolic reaction, metabolic energy of the cell, not by any kind of calcium ion across the membrane. So this is not the correct explanation. Third is, cilia is hair-like appendages. This is correct. If we are seeing the cell, the cell has very small structures. These are known as cilia that help in sweeping movement, and they are made up of composed of tubulin protein. So yes, the microtubule doublet or microtubule singlet, they are microtubules, and each microtubule is made up of tubulin protein. So the incorrect statement is B. Correct answer is B. Let's move to the next question. Next question is, which one of the following statement regarding mitochondrial membrane is not correct? First is, outer membrane resembles sieve. If we are talking about the porine protein, sieve represent the porine protein. So this can be a correct statement, because outer membrane of nuclei contain or rich in porine protein. Second statement is, outer membrane is permeable to all kind of, it is not all kind, but yes, many kind of molecule can pass through outer mitochondrial membrane. C statement, enzyme of electron transport chain या electron transfer chain are embedded in outer membrane. This is not correct answer, because enzyme present in on inner mitochondrial membrane. So correct answer is not C. So next is D is, the inner mitochondrial membrane is highly convoluted, forming series of infolding which is known as crista. So D answer is D is correct. Inner membrane have infolding, and these infoldings are known as crista or cristae. Next question is, in chloroplast which is most abundant? So the correct answer is RUBP, carboxylase. Why it is the answer? A option, phosphatase, involved in phosphate metabolism. Transketolase, it involves in pentose phosphate pathway. Which is also known as HMP pathway. D option is aldolase. Aldolase enzyme involved in glycolysis, that is breakdown of glucose, as well as glucogenolysis, that is glycogen breakdown. And B option, RUBP, carboxylase. This is present in mesophyll cell and involve in fixation of carbon dioxide, atmospheric carbon dioxide. So the correct answer is B. Next question is, organelle important in spindle formation during nuclear division is? So first option is Golgi body. So Golgi body is not the correct answer, because it involve in packaging and modification. Second, chloroplast. Chloroplast involved in photosynthesis. So how this can be responsible for cell division? C option is centriole. Yes, this is the right answer because this involves in spindle formation or formation of mitotic apparatus. D is mitochondria. So mitochondria involve in ATP synthesis. So how this is responsible for, or whenever the cell divide, mitochondria disappears. So correct answer is C. Next question is bacterial cell wall is made up of. First three options: cellulose, hemicellulose and hemicellulose. All of three A, B, C options represent plant cell wall. D option is peptidoglycan. First of all, we'll decode what is peptidoglycan. This means something is glycan means glucose plus peptido represent peptide or protein. So they are made up of glucose plus protein. That is why this is known as peptidoglycan. So we have studied that the bacterial cell wall is made up of two specific proteins, which is NAG and NAM, N-acetyl glucosamine and N-acetyl uramic acid. So these are the basic structures that are present in bacterial cell wall. Okay, then E option is glycogen. This is stored food material of animal cell. So this is not the correct answer. Correct answer is D. Moving on to the next question. Which of the following subunit of ribosome composed of 23 srRNA and 5 srRNA plus 32 different protein? So first of all, 23 srRNA is the identification feature. This is only present in prokaryote, and this is the larger or major portion of rRNA. So this portion is present in larger subunit, and larger subunit of prokaryote is composed of 50S. So the correct answer is A. While if we are talking about C option, 30S, this is the smaller subunit. This contain 30 srRNA and sum of 21 ribosomal protein. The other 70S is the full ribosome structure, so it cannot be the answer. 60S is the largest subunit of eukaryotic ribosome, and 40S is the smaller subunit of eukaryotic ribosome. So this can't be the answer. Answer is A. Next is number of protofilament present in a microtubule. So this is the direct answer. Answer is 13. D answer. Why D answer? Because each protofilament contain 13 tubulin protein arranged in a circular spiral manner. So here we are going to draw the structure. So here is the structure. They are spirally arranged and alternatively. So if we mention this alpha tubulin, this is beta. They are present alternatively, so I'll mention alternatively. Alpha. Okay, this is the structure of, and if we count this, these are total 13. 1 2 3 4 5 6 7 8 9 10 11 12 and 13. Total 13 protofilament are present in one microtubule, and these are parallel tube-like structure present in a cell. Next question is, consider following statement. Plant cell have centrioles. So this statement is utterly wrong because plants do not possess any kind of centriole. So this is wrong. A ribosome is the site of protein synthesis. B statement is correct. Ribosome are site of protein synthesis. That is why they are also known as protein factory. Next is, middle lamella is the layer mainly of calcium carbonate. No, because middle lamella is composed of calcium or magnesium pectate. Next is, in animal cells, steroidal hormone are synthesized by smooth endoplasmic reticulum. So yes, SER is responsible for lipid and lipid-like substances which are steroids, and these steroids includes androgens, progesterone, estrogen, etc., or the cholesterol like particles. If we talk about plant, so plant do not possess centrioles. So correct answer will be B and D. C is the correct answer because B statement and D statement are correct. So moving on to the next question. The number of microtubule in a flagellum, including those sharing 3 protofilament with each other, are. So they have asked about a flagellum. So these in cilia and flagella, arrangement of microtubule is 9 plus 2. 9 are the peripheral doublets and 2 are central singlet. So we are going to talk about total number of microtubule. So 9 into 2, these are peripheral, plus 2. So total is 18 plus 2, which is equal to 20. So the correct answer is C, because total number of microtubule present in a structure like flagella is 20. Okay, next is, which one of the following organism is not an example of eukaryotic cell? So first one is Euglena viridis. This is a protista, and all the protists are eukaryote. Second one is Amoeba proteus. Amoeba is also a protozoa. Protozoa belong to a class kingdom Protista, and they are also eukaryote because they have well defined nucleus. Next is Paramecium caudatum. Paramecium also belong to the kingdom Protista, and Protista all are eukaryote. The fourth option is Escherichia coli. We can write this as a E. coli. This is very most famous bacteria which is used to study the transformation, bacterial transformation. So this belong to kingdom Monera, and all the Monerans are prokaryotes. So the correct answer is D. Next are plasmid. So we have studied 3 questions today about the plasmid. These are extrachromosomal DNA which can self-replicate. Okay, this is correct. DNA carrying genetic sequence without expressing it. No, they do express it because they are responsible for unique phenotypic characters. Then C, integrated with host DNA without replication ability. So we have studied today that they have the replication ability, they have their own ori, and none of these, this can't be the option. So answer is correct answer is A, because they are small extrachromosomal DNA that have their own ori or replication site. Next is metachromatic granules. So first we'll try to understand what are metachromatic granule. Metachromatic granule is nothing, just a inclusion body or the non-living structure present in prokaryotic cell. Okay, if we are talking about metachromatic, these are polyphosphate and lipoprotein, vitamin storing structure present in various bacteria like diphtheria, bacilli, or lactic acid bacteria, also known as lab. Okay, so the correct answer is inclusion bodies. Next is chromosome with centromere situated close to its ends, forming one extremely short and one is extremely long. So the correct answer is A, because we have studied chromosome on the basis of the position of centromere. So acrocentric will look like this. One arm is very long, one arm is short. This is acrocentric. Second is telocentric. Metacentric is equal. Both arms are equal in size. And submetacentric is one arm is slightly away from the center and other arm is slightly longer. So this is submetacentric. So the correct answer is A, because they have told that centromere is situated close to the end. This is close to the end but not at the end. Here centromere is situated at the end. Okay, the next question is heterophagosome. First we'll study about the term heterophagosome. So hetero means different, and this is something food containing particle or food containing structure, we can say that. So heterophagosome is formed by fusion of few food containing phagosome with primary lysosome. So this is correct statement, because it do formed by the fusion of phagosome with primary lysosome, and we have studied that primary lysosome is something that contain inactive lysosomal enzyme. Next is newly pinched out vesicle from the Golgi apparatus fuses with endosome to become fully functional. This is something explain about intracellular transport. So this can't be the correct answer, because we are studying about digestion, intracellular digestion, not the transport. Third option is formed by fusion of primary lysosome with degenerating intracellular organelle. So this is talking about secondary lysosome, because here are degenerating organelles are getting fused. So maybe there are hydrolyzing enzyme, they are getting activated, but this can't be the correct answer, because they are talking about intracellular digestion. So this can't be the correct answer. D option, a lysosome with only indigestible food material is left. So indigestible material left structure is known as a residual body or tertiary 3-degree lysosome. So this is also not the correct answer. So the correct answer is A, because heterophagosome is made up of primary lysosome plus phagosome. Moving on to the next question. So bacteria with tuft of flagella at one pole is known as. So first we will study about all the option. Amphitrachus. Amphitrachus is the bacteria that having flagella at two ends. Second is peritrichus. Peritrichus is presence of flagella at the periphery. So number of flagella are present at periphery. Is peritrichus. Next is atrichus. So atrichus means no flagella. And last option is lophotrichus. So lophotrichus is presence of tuft of flagella at two ends. So here you should change the option from 1 to 2. So the answer will be D. Otherwise, if we talk about one pole, so the one pole at the E option, tuft of flagella at one pole, this is known as cephalotrichus. That is presence of tuft of flagella at one end only. So the answer is either cephalotrichus and, or if we change the option, so the answer will be D, lophotrichus. Then last question, axonemal arrangement of microtubule is. So let's study the structure of cilia and flagella. So cilia and flagella are covered by a single unit membrane, this is plasma, and contain 9 peripheral doublets. The peripheral doublet where A is complete, a B subtubule is incomplete, containing only 10 protofilament. We can name it as A and B. All of them are joined with each other with a layer. Each A is attached to the B and B is attached to the A of next doublet, and inner to this a presence of central sheath. Inner to the central sheath, 2 central singlet are present. These are bounded to each other through a intersinglet bridge, and this is known as peripheral doublet bridge. They contain inner radial spoke which is attached to the A subtubule, and other 3 radial spoke which are made up of nexin protein. This bridge is also made up of nexin protein, and radial spoke is also made up of nexin protein. So they have talked about axonemal arrangement of microtubule. So axonemy is, first of all we have studied what is axonemy. Axonemy is total 11 microtubule. These 11 microtubule represent 9 peripheral doublets plus 2 central singlets. So let's try to find out the answer. 6 pair of doublet and 1 pair of singlet. So this is not the correct answer. 6 pair of doublet. So here are 9 pair of doublet. This can't be the correct answer. Then next is 9 pair of doublet and 1 central pair of singlet. This can be the correct answer. Next is 9 pair of doublet and single central. There is no single central. There are double central. So we can say correct answer is C. So here are some important questions of this chapter. I hope you have got the correct answer about these questions. If you have any doubt, to let me know. Thank you so much.